Today's topic is drug-receptor interactions and pharmacodynamics. So uh, the pharmacodynamics is the main part of the pharmacology and studies the way the drugs will influence the body of the human. And there are different mechanisms for drug receptors and for the drug mechanism of action, and one of the most commonly used mechanism is receptor activation. So most of the drugs will have a specific receptor that they can activate, and once activated, the receptor will make some biologic response. And this is the most common way of the action of the drug, and most of the drugs are either agonists or antagonists of a specific receptor. And receptors can be different types. So one half of the receptors are connected with the ion channels, and these receptors are called ligand-gated ion channels. So, for example, the cholinergic nicotinic receptor is a ligand-gated ion channel, and activation of the receptor, so this part of the receptor, and this is the agonist. After the activation, that will open the ion channel that will allow the ions to freely move through this channel according to the concentration of the outside and inside of the membrane. So this is most common type of receptor. Another type of the receptor is a G-protein coupled receptor, and this is common for the adrenal receptors. And the G-protein coupled receptors are connected with the G proteins. So like these are different subunits of the G protein that and the activation of this receptor. So this is the binding site, and this is the agonist. So activation of this receptor will make the phosphorylation of the proteins that are connected with the receptor and usually are inside the membrane, and that will lead to some reaction. And then the next target of the drugs is the enzyme link receptor. So these receptors like this one, for example, will activate some reaction. So in this case, it is the phosphorylation of the molecule, and that lead to the phosphorylation of the receptor or the protein. And the last one is the intracellular receptors. So these are usually common for the estrogens, for example, and this type of the receptors are located in the cytosol of the cell, and the drug should pass through the membrane bilayer and then connect with the receptor to activate phosphorylation or the gene expression, and that is more common for the steroid receptors. So all these types of the targets will lead to the some intracellular effect depending on the type of the receptor and type of the cell. So for example, in case of the insulin receptors uh, that will increase the glucose utilization by the tissue. In case of the beta or alpha adrenal receptors, that will lead to the contraction of the muscle, and then in case of the cholinergic receptors, that probably will lead to the relaxation of the muscle, and the steroid receptors will activate some genes and produce specific proteins. And if we look more in detail to the G protein coupled receptors, so there are two types of the subtypes of these receptors, like the G stimulating and G inhibiting. So this particular is a G stimulating protein because it binds to the guanylyl D-phosphate, and the drug or hormone will activate the receptor, and that will change the conformation of this receptor, and this G protein will activate the guanylyl D-phosphate, and this will lead to the synthesis of the guanylyl D-phosphate from guanylyl triphosphate, and uh, uh, and this and after that the conformational change in the G protein will occur, and that will activate adenylate cyclase. So adenylate cyclase is an enzyme that will synthesize the cyclic adenosine monophosphate from ATP, and then so usually the increase in the cyclic adenosine monophosphate will lead to the relaxation of the muscle. So this is the way of the activation of the G protein. So like there are different subunits, so actually they dissociate in the cell membrane and then activate the some enzyme, for example adenylate cyclase. So and then when the receptor releases the uh, agonist, and then that will lead to the uh, release of the adenylate cyclase, and that will return to the previous state. So this is the way the G protein coupled receptors work. Then the insulin, the receptors, the insulin receptor uh, works another way. So the binding of the insulin activates the tyrosine kinase, and the intracellular part of the receptor activates, and that uh, leads to the phosphorylation of the uh, tyrosine that are connected to the beta subunit of the receptor, and then uh, tyrosine kinase phosphorylate other proteins, and uh, that leads to the activation of the some pathways. And then usually protein kinases and phosphatases are activated, and that leads to the actions of the insulin. So and the biologic to the some biologic response. And then the intracellular receptors are activated by the hormones. So this hormone is activates the intracellular receptor, and that receptor activated receptor goes to the nucleus and then promotes the synthesis of the activation of the specific genes, and transcription occurs, and then as the mRNA is synthesized, and then this mRNA is used to synthesize specific proteins, and that proteins will lead to some biological response. So so most of the drugs will activate receptors, and receptors can lose their activity with a repeated administration. So, for example, epinephrine is activating alpha and beta adrenal receptors, but the repeated injection can lead to the decrease of the response. So, and this this is common for the adrenal receptors. So and called the desensitization. So desensitization leads to the decrease in the activity of the receptor, but this is not constant. So after if we don't inject the drug for a quite time, and then that leads to the recovering of the response. So like this is desensitization. And to show the dependence between the concentration of the drug and the effect of the drug, the dose response curves are used for that purpose. And if we plot the concentration of the drug at the x-axis and the percentage of the maximal effect at the y-axis, we will get a dose response curves. And in these curves, we are interested in effective concentration 50. So effective concentration 50 is the concentration of the drug that will lead to the 50% of the maximal effect. So first we plot the graph and then find the maximal effect of the drug. So this maximal effect is usually in the high concentration. So if we increase concentration further, the effect will not increase anymore. So this is a maximal concentration that is considered 100%. So then we take a half of this concentration and plot a line, and then at the point it will cross the graph, we will plot a perpendicular line, and then we will get a EC50. So if we make the same graph with the logarithmic concentration of the drug, then it will the graph will change its shape to the S curve. So like if this is a hyperbola as the normal linear graph, and then at the logarithmic scale that will be S like curve, so that allows more accurate detection of this EC50, because now EC50 is actually will be the half of the graph. So, and logarithmic scales are used for that purposes. And the lower is the EC50, the more potent is the drug. So the potency of the drug is measured with the effective concentration 50, so half effective concentration. and the efficacy of the drug is measured by the biological effect. So for example, here we have 3 drugs, and drug A is more potent than the drug B because the EC50 is lower of the drug A is lower than the EC50 of the drug B, but they have identical biological effects, so efficacy of the drug is the same. And then drug C has even lower potency because EC50 is even higher, and also because biological effect is lower, and it also have lower efficacy. So drug C is actually less effective and less potent than drug A and drug B, and drug A and drug B are equally effective, while drug A is more potent. So for the clinical purposes, the effectiveness of the drug is more important than the potency, because potency is only only shows the concentration of the drug, and the concentration is usually prescribed according to the requirements, so that is not a problem to increase the concentration, provided the effect is there and there are no adverse effects. So this actually, the 2 graphs show the response of the drug to the depending on the dose. So with the increase of the dose, the binding to receptor increases until all receptors are bound at this maximal dose, and then that is why the dose will not increase further. So that means that the amount of the receptor is limited, and that is why we cannot overcome this limit. So, and the same is for the logarithmic scale, and the logarithmic scale actually shows the same, but the curve is S-like. So then if we use a antagonist, so we have actually full agonist and partial agonist. So difference between the full agonist and partial agonist is that the full agonist will produce the maximum response at 100%, while the partial agonist will only show lower response than the full agonist. So even in the highest concentration, it will not make 100% effect. That is why if we add a partial agonist, so if the patient takes full agonist and then we give him partial agonist will decrease maximal response and will act like an antagonist. And then there is also inverse agonist that will decrease the response from the basal level. So like when there is no drug at all, still there may be some physiological response, but inverse agonists will decrease it further. And most of the drugs are competitive antagonists. A competitive antagonist means that the drug and receptor will the drug and the agonist will compete for the receptor. So usually the agonist is synthesized in the body, and the antagonist is a drug. And then if we have this response for the drug alone, so the antagonist, the competitive antagonist, will decrease this response, so the maximum effect will be lower because part of the receptors are connected with the antagonist and part of the receptors are connected with the agonist. So with the sorry, this was non-competitive antagonist. So like the, the drugs will not the agonist will not reach the maximum effect with the non-competitive antagonist, because a non-competitive antagonist will not release with the connection with the receptor. So once the non-competitive antagonist is bound to receptor, it cannot compete with the agonist. While with the competitive antagonist, so there is a specific shift to the right. So the dose response curve is moved to the right, and that means that the the agonist can still uh, bind to the receptor, but higher dose is required for that because the receptor is occupied with the drug, but it can be released, antagonists can be released from the drug, uh, from the receptor, by the higher concentration of the agonist. So most drugs are actually competitive antagonists, and that makes them safer because in case of the overdose, we can just stop the administrating the drug, and then it will release from the receptor itself. With the non-competitive antagonist, that is not the case. So and more safe drugs usually have a large therapeutic index. So therapeutic index is another safety um, criteria, and if the therapeutic index is low, so therapeutic index is actually the difference between the toxic doses and the therapeutic doses. So and if the difference between the uh, therapeutic doses, that is commonly called therapeutic window, uh, are not much different from the adverse effect concentrations of the drug, then, and sometimes they are overlapped like here, then adverse effect can occur with the high doses. So this is called a small therapeutic index. And then with a large therapeutic index, the adverse effects are usually occur at much higher doses, like 5 times larger than the therapeutic effect occurs, and that uh, and that says and these drugs are usually safer. Like for example, penicillin is a uh, antibiotic uh, that is most safest antibiotics, and the group of the penicillin is one of the most safest antibiotics. So and warfarin is actually is an anticoagulant, and it is has a small therapeutic index, and that lead to the overdose and bleeding, commonly encountered bleeding during the treatment because the therapeutic index is low. And that is it for today. Thank you for your attention. And do you have any questions?
No, thank you.
Uh, so if no question, then see you next week, uh, same time. Bye bye.
Thank you, Doctor. Goodbye.